Hello, my name is Niveda. I'm a PhD student in the lab of Professor Junaki Singh in the Biological Sciences and Bioengineering Department at the Indian Institute of Technology, Kanpur. We have recently published our work in the February 2023 issue of the scientific journal Development. In the video that follows, I will explain what our research has revealed. So, let's get started.
A group of scientists at the Indian Institute of Technology, Kanpur, who study brain hemisphere formation using the developing chick embryo as a model organism. One might ask, how are the brain hemispheres formed during development? I will explain this process simply by imagining the brain at the very beginning to be a round water balloon. Below the analogy, the events happening in the chick brain is shown at the same time. As the embryo develops, the top of the balloon is being pushed, forcing it to fold inwards. Further in development, this fold deepens, separating the round water balloon into two hemispheres. An obvious question here is, what happens to the brain if the top doesn't fold inwards? In that case, a severe developmental brain disorder called holoprosencephaly occurs, and that is why it is important to study this inward folding process of the top of the brain. The main question that we have asked in the study is that, what are the molecules working behind the scenes that make the top of the brain to fold inwards? To date, we only know of one molecule called retinoic acid, which is formed from vitamin A that is crucial for this inward folding at the top of the brain. We did this study to find what genes and molecules, apart from retinoic acid, can drive this inward folding, ultimately forming the brain hemispheres. And what we found was very interesting. We started by searching the inward folded region for new genes or molecules that could possibly be involved in this folding process of the brain, and that is when we found a new gene called CNKSA2 in this region. So we questioned how is this gene involved in the development of the chick brain. To answer that, we removed CNKSA2 from the inward folded region of the brain by a technique known as RNA interference, where the RNA formed from the DNA is destroyed. When we did this, we found no folding at the brain top as well as no hemisphere formation. This led to an abnormal brain similar to holoprosencephaly. But the question is, why should removal of CNKSA2 do this? We found that CNKSA2 works in hand with another important signaling pathway called the Ras-RAF-MEK signaling. In the normal scenario, CNKSA2 is present at high levels in the inward folded region of the brain, while the Ras-RAF-MEK signaling is low, allowing the top of the brain to fold inwards and form hemispheres. On the other hand, removal of CNKSA2 from the top folded region causes the Ras-RAF-MEK signaling to shoot up, and that is why the top of the brain cannot fold and separate into the two hemispheres like that of what is seen in holoprosencephaly. Therefore, in conclusion, through our study, we have found and added new players involved in the inward folding process of the brain and eventually in brain hemisphere formation. The importance of these findings are that women in the childbearing period may be advised to avoid taking anything through food or in medicine that might disrupt the separation of the cerebral hemispheres in the baby. Anyone interested can read the full research article published online.